Hi everyone. In today's video, I wanted to go through the four main membrane receptors, and this is associated with the process or the concept of the endocrine system, and is actually also associated with pharmacodynamics, as different drugs interact with different receptors in order to have different effects. So the first receptor that I'd like to go through today is called a ligand-gated ion channel. Now, if you're seeing this word for the first time, it can be a bit daunting, but once you break it down, it's quite easy to actually understand. So first, let's go through the term ligand-gated. Ligand-gated basically means that a certain molecule, called a ligand, is able to bind to a receptor in order to cause for a conformational change or a shape change in the receptor, and therefore allow for a particular effect. Now, specifically, a ligand is a molecule that is a chemical messenger, so a hormone, a gas, or any other type of messenger, chemical messenger, that is able to bind to a receptor in order to have this particular effect. So examples of ligands can be neurotransmitters, such as serotonin, steroid hormones, such as testosterone, and even pharmaceutical drugs. So, now let's explore the term ion channel. Ion channel basically means, if you've seen my video on membrane transport already, a channel that allows for the passage of ions from either the inside of the cell to the outside of the cell, or from the outside of the cell to the inside of the cell. So allows for passage of ions. So let's take an example of this. If you're familiar with the nervous system, you would actually know that we need an action potential in order to allow for the formation of a nerve impulse. But how is that particular action potential started? Well, we actually have in the synaptic cleft, which is a space between two neurons, a ligand-gated ion channel. Say, for example, this is the bilayer of the postsynaptic neuron. A ligand-gated ion channel would essentially have a structure similar to this, and if there is no ligand bound to it, it would be closed. Now, say for example, a particular neurotransmitter, say serotonin, is able to activate this ligand-gated ion channel in order to incite a particular response. So serotonin will pass from the synaptic cleft towards the ligand-gated receptor, and it will bind to it. This binding, as I mentioned earlier, will have an effect that will change the conformation of the ligand-gated ion channel. In this case, it may allow for the two subunits of the ligand-gated ion channel to move further apart from one another. And as they move further apart from one another, sodium ions will be able to pass through. Therefore, the binding of this particular neurotransmitter has allowed for the ligand, for the ion channel, to change shape. And when this neurotransmitter is removed, Then the ion channel can return to its original shape and therefore wait for the next neurotransmitter to allow for ions to pass through. The next type of receptor that I wanted to go through today was a G protein coupled receptor. Now, these receptors were actually a little bit more complicated than ligand-gated ion channels. Firstly, ligand-gated ion channels are very fast-acting and can have an effect within milliseconds. G protein receptors, on the other hand, have an effect that can come about around in a couple of seconds, which means that they are 1,000 times slower than ligand-gated channels. So how does the G protein coupled receptor work? I think in this case it's best to represent it diagrammatically. So if this represents the lipid bilayer of a particular cell, a G protein coupled receptor will have a structure in which there is a receptor, and the actual structure of the G protein will pass through the membrane of the cell 7 times. Now, this is a unique structure of the G protein coupled receptor and allows for them to be easily identified within cells. So what I mean by the coupled receptor passing through the membrane 7 times is that, similar to alpha helices, it will pass through once, then twice, then thrice, then 4 times, 5 times, 6 times, and 7 times. If you have trouble remembering this, then I like to think that as G is the 7th letter of the alphabet, it will pass 7 times. So, what happens on the other side of the cell? If this is the extracellular environment, and this is the intracellular environment, then what actually happens is that the G protein coupled receptor has what we call an alpha beta gamma complex. The alpha beta gamma complex is quite important, as it is essentially what will allow for the activation of another enzyme or any other process within the cell. So, this alpha beta gamma complex, what does it mean? The alpha beta gamma complex is essentially what we call the G protein. So, it'll be structured something like this, with the gamma substructure here, the beta substructure here, and the alpha substructure here. Now, this receptor is activated by a particular chemical messenger binding to its receptor that is located outside of the cell. So if we have a random chemical messenger here, denoted by M, if it binds to this receptor, then what will happen is another conformational change, similar to the ligand-gated ion channels. The conformational change in this place will actually be the separation of the alpha subunit from the beta and gamma subunits. This will essentially mean that the alpha subunit will come detached and will maintain its adherence to the lipid bilayer and will move away from the G protein. So, as the alpha subunit moves away, what is actually providing this driving force for it to move away? Well, essentially, when the alpha subunit, if we take a zoom in perspective here, when it is inactivated, it actually contains a GDP molecule attached to it. And if you've heard of all the triphosphate molecules, like ATP, you'll notice that it's simply just a different nucleotide to ADP, adenosine, Instead of adenosine, it's guanine. So it serves a similar process. What the activation process does is that it swaps this GDP molecule from the alpha subunit and replaces it with a more energetic GTP molecule. This GTP molecule, within its phosphate groups or phosphate bonds, is able to provide energy for the process of the movement and activation of a particular enzyme. So say, for example, we have a particular enzyme present here. And in order for it to be activated, the G protein or the alpha subunit of that G protein must bind to it. So as the alpha subunit moves along the lipid bilayer being attached to it, it will eventually bind to this enzyme. And the binding of this enzyme to the alpha subunit will activate it. Therefore, a particular cellular response will be brought about. So this is basically the action of a G protein. It starts off with a receptor binding to the protein, then allowing for the conversion of GDP into GTP on the alpha subunit, allowing for the alpha subunit to therefore move away from the beta and gamma subunits and bind to a particular enzyme in order to activate it. The next type of receptor that I'd like to go through is called a kinase-linked receptor. Now, unlike ligand-gated or G-protein coupled receptors, kinase-linked receptors are actually quite slow. They usually take around an hour to insert their effects. So what does a kinase-linked receptor actually entail? Well, similar to G-protein and ligand-gated ion channels, these receptors are extracellular receptors. This means that they contain receptors in the extracellular environment. So say, for example, we have a particular receptor that is shaped as so. A ligand-gated receptor is unique in that it actually contains two binding sites. And in this case, a particular chemical messenger that is shaped complementary to the receptor is able to come forward and cause for a conformational change. This conformational change is what eventually will allow for the effect to be incited within the cell. So as you can see, the two receptors or the two subunits of the receptors actually come closer to one another. This nature of coming closer is actually very important, as it essentially allows for the effect to be seen within a cell. So how do these membrane receptors actually coming together help us in the process of what we want to achieve? So essentially, these two subunits that are present on the kinase-linked receptor actually contain many tyrosine groups. Now, as we all know, tyrosine is an amino acid. Now, what actually happens when these two groups are brought together is that these tyrosine groups are phosphorylated. This means that a phosphate group is added to them. This is a very important stage, as it essentially means that the receptor or the extension of the receptor into the intracellular environment, is able to act as a docking station for particular enzymes. And there are two main ways in which these enzymes can actually attach to these receptors. So firstly, an enzyme that has the complementary shape to the particular phosphate group can directly bind to it, as so, and then it can have its particular cellular response. Whilst on the other hand, if a particular receptor or enzyme, is not complementary, then we have what is called a scaffolding protein. Scaffolding proteins or scaffolding enzymes are essentially enzymes that are able to bind to this kinase link receptor, and the other enzyme that is actually meant to bring about the response within the cell is able to bind to this scaffolding protein in order to incite a response. So what do I mean by that? So if this is the particular enzyme that we will know will incite the particular response within the cell, but it doesn't fit onto the phosphate groups, then a scaffolding enzyme, which is able to bind to the phosphate groups, can come in. So I'll just redraw this. And therefore, it'll bind. And this enzyme can come about and bind to the scaffolding enzyme to therefore incite its own response. So these are the two ways in which a kinase link receptor can allow for a particular cellular response. The final type of receptor that I wanted to go through today is called an intracellular receptor. Now these receptors also have another name, in that they are also called nuclear receptors. Now why is this the case? Well, essentially, the three receptors that we've looked at so far, ligand-gated, G-protein, and kinase link receptors, have all been receptors that have their receptor sites on the outside of the cell. Intracellular receptors, on the other hand, have their receptors in the nucleus, or somewhere inside the cell. This is seen in receptors such as thyroid hormones, which are able to be transported in by carrier proteins in order to reach the nucleus, or by steroid hormones, which are able to directly enter the cell as they are lipophilic and therefore can pass through the lipid bilayer. These receptors, like kinase-linked receptors, are also quite slow and therefore require hours for their particular response. Let's look at an example. Say this is the lipid bilayer of a cell with the intracellular environment and extracellular environment. And down here we have the nuclear membrane, this side being the cytosol, and this side being just the nuclear plasm, which I'll abbreviate to NP. So if we have a particular intracellular chemical messenger, such as a steroid hormone, then there are two main ways in which the steroid hormone can have a particular effect on the cell. Firstly, the steroid hormone can enter the cell, or if it's a thyroid hormone, can enter through a specific carrier protein, and it can bind directly to a receptor within the cell. So that we have the steroid hormone here binding directly to a receptor, and this receptor can take the hormone or allow for its particular response to occur within the nuclear membrane. And this effect can be, for example, on the genetic material which is present in the cell, and that's why this process takes hours, as it can allow for the process of transcription and translation to occur. And as these processes are involved with the genetic material of the cell, they're quite important and therefore must be checked off quite stringently. The second way in which a particular chemical messenger can have an effect on a cell is by directly moving into the nuclear membrane, into the nucleoplasm, and directly interacting with the genetic material in the cell. This means that they are able to directly allow for the processes of transcription and translation to occur. This can bring about a variety of different effects on a particular cell. So, in review, the four types of chemical receptors that I've explored today are ligand-gated ion channels, which are defined by a chemical receptor opening a channel to allow for the flow of ions through it. A G-protein coupled receptor, which involves three subunits which are able to detach and allow for the activation of an enzyme with the alpha subunit. A kinase-linked receptor, which allows for a chemical messenger to bind to two subunits of the receptor to bring them closer to one another, and therefore allow for the phosphorylation of different enzymes to therefore allow for their activation. And also intracellular receptors, which are able to allow for the change in genetic material or any other process within the cell by entering the cell and not interacting with its extracellular environment. Now, just before I finished, I quickly wanted to go through what can happen with a particular receptor when something goes wrong. And the example that I wanted to take here was with G-protein coupled receptors. These receptors are very important within the body as they are essential for the control of different fluids. So two examples of pathogens that I wanted to explore today are whooping cough and cholera. Firstly, whooping cough. Whooping cough is a condition that arises when essentially there is too much of liquid or a particular substance such as mucus within the lungs that builds up and essentially causes for a wet, hacking cough to result in the patient. So how does this actually relate to G-protein coupled receptors? Well, G-protein coupled receptors are actually really important in the lungs as they're responsible for the entry and exit of particular liquids within a cell in the lungs. So what essentially happens in whooping cough is that the bacteria, which is called Bordetella pertussis, is able to interact with cells in the lungs. And let's say this is the cell. And this cell has a G protein coupled receptor, 1, 2, 3, 4, 5, 6, 7, present on its extracellular surface. And essentially what happens is that there are two types of G protein coupled receptors, inhibiting receptors and stimulating receptors. But in this case, in the inhibiting receptor by the Bordetella pertussis bacteria is activated, which essentially means that the effect of this receptor, which essentially allows for the formation of a molecule called adenylyl cyclase, is not controlled because this chemical messenger is permanently bound to this protein. And the overaccumulation of this particular molecule means that fluid that is already maybe present outside of the lungs is unable, or sorry, not outside of the lungs, in the lumen of the lungs, is unable to pass into the cell. This essentially means that fluid builds up within the lungs and causes for that wet hacking cough. Cholera, on the other hand, happens in the digestive tract and involves a similar mechanism, but instead of the inhibiting G protein coupled receptor, it involves the stimulating one. So, in this case, the cholera bacteria acts as a chemical messenger, binds to the stimulating G protein receptor, and it essentially allows for the buildup of a molecule called cyclic AMP within the cell. This enzyme is called cyclic adenosine monophosphate. And unlike whooping cough, which involves fluid that is already present within the lungs not being able to pass into the cells and therefore be drained, fluid within the cells or from other parts of the body is able to seep out uncontrollably from the cells. This loss of fluid is what causes, and as this occurs in the digestive tract, is what causes for excessive diarrhea in patients, as essentially there is a lot of fluid going into the digestive tract of these patients, and in order to get rid of it, there's really only one way, is to get rid of it outside of the body. The effect of this is also that as a lot of this water has been taken out of different parts of the body, it can result in dehydration. So this was just a little bit of an insight on the 4 different types of receptors that exist in the body, and just 2 small examples of what can go wrong with one of these receptors. Thank you for watching.